Okay, well, we're ready to shift gears now and talk about our metabotropic receptors. So again, these are receptors for neurotransmitters that are not ion channels. Rather, these receptors interact with a series of second messenger pathways within cells that lead to a variety of postsynaptic effects. Perhaps the opening and closing of ion channels, but maybe a great diversity of other kinds of postsynaptic effects as well. So here's a typical example of a metabotropic receptor, also known as a G protein coupled receptor, because the receptor itself is a monomeric integral membrane protein that is associated with G proteins on its cytoplasmic surface. When the receptor is activated by the neurotransmitter, these G protein systems then become active. And those G proteins then can interact with effector proteins, and those effector proteins can produce a variety of intracellular messengers that potentially can gate some ion channel. So in this example, an ion channel does indeed open following the binding of a neurotransmitter. So between the binding of the transmitter to a receptor and the opening and closing of an ion channel in this example are a series of metabolic reactions, and that takes time. So the metabolic steps are what accounts for the slower action of metabotropic receptors on postsynaptic conductances relative to our ionotropic receptors, where the effects are essentially instantaneous with the binding of a neurotransmitter to its receptor. So let's look at a couple of different examples. So the receptor itself is typically a monomeric integral membrane protein, but the intracellular second messenger systems related to G proteins can be quite diverse. We have our more canonical heterotrimeric G protein systems illustrated to the left, and so activation of G proteins alpha as well as the beta and gamma G proteins can go on and mediate a variety of postsynaptic effects interacting with effector proteins, some of which may produce second messengers themselves, others which may activate enzymes that can lead to the phosphorylation of target proteins and the activation of a variety of systems within postsynaptic cells. Other receptors for neurotransmitters are associated with monomeric G protein systems, such as the RAS system. And activated RAS can go on and mediate a variety of postsynaptic effects itself. Here's some specific examples. Are receptors for many of the biogenic amines, such as dopamine, norepinephrine, serotonin. They interact with G protein coupled receptors. Illustrated to the left is the beta adrenergic receptor for norepinephrine. It activates a GS version of the heterotrimeric G protein system, which leads to the activation of adenylyl cyclase and an increase in the amount of cyclic AMP that's produced within the cytoplasm. Cyclic AMP is an important second messenger system that can then lead to the increased phosphorylation of target proteins. Glutamate binds not only to the ionotropic receptors like the AMPA and NMDA receptor, but also to metabotropic receptors. Here, glutamate is binding to a receptor that activates GQ proteins, and these can lead to the activation of phospholipase C and the activation of other kinds of second messenger systems, such as the IP3 pathway and the diisoglycerol pathway, again leading to the phosphorylation or perhaps the dephosphorylation of target proteins within the cell. And then lastly, we see that dopamine binds to metabotropic receptors. We'll talk more about this when we talk about the circuitry of the basal ganglia. Dopamine is binding to a D2 receptor here, which interacts with a GI protein, in this case leading to the inhibition of adenylyl cyclase and a reduction in the levels of cyclic AMP. And that leads to a decrease in protein phosphorylation in the target site. Now, one benefit of the G protein-coupled receptor family is that there can be tremendous amplification of the signal as these metabolic steps play out. So imagine activation of a single receptor leads to the production of a fairly large number of G proteins. So there's amplification in this step. Well, in the pathway, for example, following beta-adrenergic receptor activation, these G proteins can activate adenylyl cyclase enzymes, which then produce cyclic AMP. So another significant amplification step happens where a large number of cyclic AMP molecules are derived from the activation of a smaller subset of adenylyl cyclase molecules. These cyclic AMP molecules then can interact with protein kinases, which leads to the massive phosphorylation of numerous target proteins. So significant amplification is the end result of activating a G protein-coupled receptor. G protein-coupled receptors can not only activate conductances in the postsynaptic cell, they can also interact with transcription factors that can modify gene expression. And these kinds of interactions mediate the longest lasting effects of G protein-coupled receptor activation. So there are pathways that follow the activation of heterotrimeric G protein systems that lead to the activation of regulatory elements such as the Krebs binding domain in our genes. And activation of Krebs can lead to modification of gene transcription. Similar Krebs modifying pathways can converge upon the same machinery within the nucleus through different pathways. Here's an example of a RAS-mediated pathway also activating Krebs. Sudden rises in intracellular calcium likewise can interact with calmodulin-dependent kinases to activate Krebs. So there may be a variety of ways that we can influence the transcription of the genome based on the interactions that are happening out at the plasma membrane of a neuron. And G protein-coupled receptors are one important means for doing so. If we look at the molecular structure of G protein-coupled receptors, what we find are typically 7 transmembrane domains. These 7 transmembrane domains are characteristic of this gene family. And so we now know that they encode a variety of G protein-coupled receptors that show up in some surprising places. So what they have in common is that they are a monomeric protein. There is an extracellular domain, which typically includes an N-terminus and a site for the binding of a neurotransmitter. In this example, what we're seeing is the muscarinic variety of acetylcholine receptor. There is, of course, a cytoplasmic side of the molecule that interacts with the G proteins. So we expect to see that as well. And again, the defining feature are these 7 transmembrane domains that run through the plasma membrane. So, as I said, there are a variety of G proteins, and they show up in some surprising places. We'll find G proteins, for example, involved in sensory transduction in some of our special sensory systems, including the olfactory system and even the visual system. So the transduction of photons into electrical energy requires the activation of a G protein-coupled receptor system. So there's a variety of metabotropic receptors for a large family of small molecule neurotransmitters, as well as for our peptide neurotransmitters. And the complexity of this system is just now beginning to be appreciated. What's important for you to take away is the idea that the metabotropic receptors give rise to these more slowly evolving but potentially much more long-lasting effects of the binding of a transmitter to its receptor. So now that we've talked about these diversity of receptors that are present that interact with these neurotransmitters, I think you can understand why we cannot judge the physiological effect of a neurotransmitter by only knowing something about the identity of the transmitter. and the kind of chemical structure of that transmitter. No, that's not enough. We have to know about the receptor in order to understand the physiological effect. And that receptor might be an inotropic receptor in mediating a fast effect on a postsynaptic conductance, or it might be a metabotropic receptor in mediating a slowly evolving effect that impacts a postsynaptic conductance but could also alter gene transcription and thereby have a long-lasting effect on that postsynaptic cell. Well, we're going to come back to some of these themes, and we will return to our metabotropic receptor family. As I mentioned, they show up in some pretty surprising places. We'll discover G-protein coupled receptor systems in our special sensory pathways, such as our olfactory system and in our visual system. And perhaps you'll be surprised to discover that the act of seeing requires the activation of a G-protein coupled receptor, very much like the same kinds of receptor structures we've just been looking at. So stay tuned for that. But meanwhile, now that we understand some of the basic molecular and cellular mechanisms that are responsible for chemical neurotransmission, we're ready to talk about the integration of electrical signals in a postsynaptic cell, and then that will allow us to talk more about synaptic plasticity and how the effectiveness of a synapse can be altered by its very use. So I look forward to having those discussions with you in the next few tutorials.